Okay, let's go over the overall process of photosynthesis. Before we go through this crazy diagram, I wanted to kind of take you back to this one first. So I want to point out that this process of photosynthesis, if you look at this diagram, what this membrane right here is showing you is the actual thylakoid membrane. So this stroma that it's showing us out here and this thylakoid space, if I go back to the diagram I looked at earlier, the stroma is kind of like this open space outside these thylakoids. So when I'm looking at this kind of thylakoid taken out, this membrane of this individual stack is where this entire process is taking place. So to start this out, number one, I want to point out this is photosynthesis we're talking about. So this is happening in a chloroplast in a plant cell. You're not doing photosynthesis because you're not a plant. So to start off the process, as you know, photosynthesis involves light. So you're going to start off right over here. Light is going to strike photosystem 2. What a photosystem is essentially a chlorophyll molecule. So you can't really see it very well in this diagram, but you're going to have small little chlorophyll molecules and receptor molecules along that thylakoid membrane. What those are called or what the specific names are, you don't need to know. You just need to know that it's called a photosystem. There are actually two photosystems here. Light's going to strike photosystem 2 first. I don't know why 2 comes first, but it does. When light strikes photosystem 2, what happens is a water molecule inside the thylakoid is going to split. So when water splits, it's going to split into hydrogens and oxygens. So your hydrogen is going to then donate its electron to the photosystem. So water's going to split. You have 2 hydrogen ions, each donating their electron to this electron kind of chain that's going to take place. The oxygen is going to actually leave the chloroplast by diffusion because you're going to have a lot of oxygen inside, so you're going to build up a gradient. It's going to want to leave. And as you know, plants give off oxygen that we need. So essentially what's going to happen is your electron is going to get transferred here. That energy of that moving electron is actually going to be used to pump, and we'll talk about where this comes from later, another hydrogen ion from the stroma into the thylakoid space. Now, this is opposite the gradient. So this is actually what we call active transport. You learned last week that involves energy. Now that energy that we're using is actually this movement of that electron. So it's almost like you're using the kinetic energy to pump that hydrogen ion in. Then what happens is essentially we kind of take the energy away from the electron. So light is going to then strike again, photosystem 1 this time, which is going to re-energize this electron to continue through this pathway. So once the electron gets through the pathway, it's going to get kicked out into the stroma where it's going to join up with a hydrogen ion. So the first part of the system, just to kind of review this again, light strikes photosystem 2, a water molecule splits in half. It forms hydrogens and oxygens. The hydrogens donate their electrons to the photosystems or to the electron transport chain. You're going to pump that electron through these carrier molecules. It's going to pump a hydrogen ion into the thylakoid from the stroma to the thylakoid, which de-energizes your hydrogen ion. Light's going to strike photosystem 1, re-energizing your electron, allowing it to get kicked out into the stroma where it's going to meet up with a hydrogen ion. Those hydrogen ions that are pumped in stay inside the thylakoid, and your oxygen is going to leave by diffusion. So that's the first part. Now, looking at this membrane, you're going to see this weird, funky looking, kind of almost like a guitar-shaped thing. This molecule is called ATP synthase. This is an enzyme that's built into this membrane. Its job is to create ATP. So synthesis, means to build. The ending ase means an enzyme. So ATP synthase is going to help build ATP, which we learned last week was our energy molecule. So basically what happens is all of these hydrogen ions that have built up within this space are going to want to naturally go through, they want to get out. We're building up a high concentration here, so we're forcing them to come through this molecule. As they come through this one particular channel, what happens is ADP is going to be converted into ATP. And you notice these kind of big chain thingies here happening, and I'm going to go over those with you in just a second. So ADP, ATP, that hydrogen then gets kicked out and is just kind of hanging out here. Now what's going to happen with this hydrogen ion is you'll see it's right next to where that electron is coming out. So it's going to hook up with its electron and form a regular hydrogen molecule. So the other piece of the puzzle from the first part is this electron. So you'll notice we have this NADP, NADPH thing happening. It's almost like ADP and NADP are kind of taxis of the cell. So ADP is going to pick up a phosphate, it's going to form ATP, it's going to take that energy, because that's what that is, right? It's carrying energy up for this next process. It's going to drop off the energy, the phosphate's going to disconnect, and it's going to then come back down to pick up another phosphate and more energy. So you can imagine this is almost like a little loop that it's going on. It's picking up a phosphate and energy, it's taking up here, it's dropping off the energy of the phosphate, it's coming back down to get re-energized. So it just stays on that track nonstop. The same thing is happening over here. NADP is going to come, it's going to pick up that hydrogen molecule. Remember, hydrogen came through, it hooked up with our electron, it hooks onto this molecule. Then NADP becomes NADPH, carries that hydrogen up for this process, drops off the hydrogen, comes back down and picks up another one. So NADP stays on this track where it just becomes a hydrogen taxi. So both of these, without both of these molecules, this next part, which is where we're actually building the glucose, wouldn't happen. So up here at the top, this is called Kelvin cycle. And in Kelvin cycle, essentially what you're seeing is this is where we finally get the CO2 that you know that plants come in. So it's going to diffuse into the cell, it's going to diffuse into the chloroplast. That 6CO2 is going to break apart into, it's going to kind of rearrange itself into what we call 3 PGA molecules, which, like, you know, the Professional Golf Association, that's the way to remember that. What's going to happen is we're going to give some energy to those molecules, we're going to add in some hydrogens, and we're going to form what we call G3P. So we go from CO2 to PGAs, we give it energy and hydrogens, and we form G3P. Now, I just am going to stop for just a second and point out there are some crazy complicated chemical things happening up here, and you don't need to know them. What happens then is we have to go through this process twice in order to form glucose. So essentially what G3P is half of a glucose molecule. What we have when we go through a molecule, we form a G3P, we have some stuff left over. Even after we go through it twice, so after 2 turns of this, we make a glucose molecule. It's going to stay in the stroma, it's going to form a starch molecule. But you kind of have some left over, and that's where this guy Rubisco comes in. Rubisco is just an enzyme that kind of rearranges everything and gets it ready to go around again. So again, this is Kelvin cycle, which is the process that happens in the stroma of the chloroplast that actually makes your glucose.